Okay, great. I hope everyone can hear me. So my title is Tumor Immunity at Single Cell Resolution, but I'm going to talk about a lot of methods today, and I hope that's okay. We've been doing a lot of methods recently. I'll start with a more biological story about engineering immunogenic cell death to enhance immunotherapy. This is a project with a cell death expert, Emily Cheng, as well as Ming Li, an immunologist. But I'm also using it as a showcase of how you can use topic models to understand single cell RNA in the immune response. And then I'm going to quickly present two different sort of methods, projects. One is on automating annotation of the tumor immune microenvironment when you do single cell RNA profiling of patient tumors. And then the last, as I'm probably going to be running out of time, I'm going to talk about cell space, which is a new sequence-informed embedding algorithm for single cell attack that I'm quite excited about. Okay, so we've spent quite a bit of time thinking about what are the limits to immunotherapy. And I know Yuri already presented his paper. I'm just summarizing here that we all know that the idea behind immune checkpoint inhibition in immune checkpoint inhibitors is to exploit the presence of tumor-specific CD8 T cells and rescue their function by using blocking antibody against inhibitory molecules that are expressed either by T cells or tumor cells or antigen-presenting cells. And there's an emerging consensus from the last 5 years. Maybe immunologists know this, but I'm always surprised in cancer audiences that people don't know about this emerging consensus that most tumor-specific T cells actually don't respond to immunotherapy because they're in a terminally differentiated, non-responsive or exhausted or dysfunctional state, as Yuri already talked about. And in fact, it's very impressive how their chromatin accessibility state doesn't budge under immune checkpoint blockade. There's a small population of progenitor cells that do respond to immune checkpoint inhibitors called progenitor dysfunctional cells. At least early after antigen presentation, these progenitor dysfunctional cells are very similar to memory precursor cells. And as Yuri mentioned, there's a common trajectory in tumors and chronic infection establishing this terminally dysfunctional state. And this is work of many labs. I'm just citing 2 papers that we were involved in. Okay, so I'm going to talk about a different concept for how to think about the limits of current immunotherapy. And in particular, often immunotherapy doesn't work as a monotherapy, right? So in some tumors, there is some efficacy as a monotherapy, melanoma, clear cell renal cell carcinoma, non-small cell lung cancer. But even there, even in the most responsive tumors, most patients don't achieve complete durable responses. Okay, and in many cancers, there's no efficacy at all, or little to no efficacy. So there are many efforts to try to combine traditional therapies, chemotherapy and targeted therapies with immune checkpoint inhibition. Perhaps we have to step back and realize that many of these traditional anti-cancer therapies are actually killing cancer cells in the wrong way, okay? And so this is a picture from Emily Cheng's lab, put this together, right? So most anti-cancer agents kill cells through apoptosis, and this is immunologically quiet, okay? And what happens, you can initiate this cascade with truncated bid. It's back dependent. You have outer membrane permeabilization for the mitochondria and release of cytochrome C, and then you have this apoptotic cascade. And if some mitochondrial DNA leaks out of the mitochondria, the apoptotic cascade actually inhibits C-gasting. Okay, so that's, if you can see my cursor here. Okay, so what the cell death field has recently realized is that if you can activate back in the absence of the apoptosome, you actually get this kind of intermembrane permeabilization, efflux of mitochondrial DNA, cytoplasm, and if you can shut off this apoptotic cascade, you get an induction of C-gasting. It should be very immunogenic, okay? And this cell death mechanism, my collaborator has coined MIMPtosis, okay, for this inner membrane permeabilization. Okay, so we have an immunogenic cell death, MIMPtosis, and a non-immunogenic cell death, apoptosis, okay? And just a little bit more of the kind of functional data, what Emily Ching's lab did was engineer this well-used B16 melanoma model with TET-inducible truncated bid, and in both wild type and APAF1 knockout. Okay, so in the knockout, if you give doxycycline, you induce T-bid, you get MIMPtosis. In the presence of APAF1, you get apoptosis. All right. So in this experiment, this is a re-challenge experiment. So you inject the engineered B16 cells, you let the tumor grow, then you give dox, the tumor regresses, you have these different cell death mechanisms, and then you inject the B16 cells on the other side, and you see, is there a memory response, okay? Or did the mice get melanoma and die? Okay, and what you can see is that this MIMPtosis cell death actually protects the mouse from rechallenge. Okay, I'm not talking about in the paper. We actually have apoptosis, MIMPtosis, necroptosis, and pyroptosis. I'm not going to talk about the other cell death mechanisms. Suffice to say that MIMPtosis is the most immunogenic cell death. It confers memory against rechallenge. And I'll also say that this APAF1 is sometimes lost. Its expression is sometimes lost in melanoma and other cancers, for example, through DNA methylation. So it's sort of relevant, this actually happens in tumors, and for those tumors, you can think of targeting them. Okay, so where we came in was we wanted to understand what happens when you initiate this immunogenic versus non-immunogenic cell death, what happens in the immune response. And so we did single cell RNA, also actually parallel TCR sequencing at an early time point. Okay, so you inject these engineered B16 melanoma cells, wait for tumors to grow. Then you give doxycycline, so you're initiating apoptosis or MIMPtosis, and at time 0 or time 48 hours, let's see what happens. Okay, so at a broad scale, it's not so hard to analyze the major clusters, to annotate what's going on, okay? And I'll just sort of, at time 0 in either apoptosis or MIMPtosis, you mostly have cancer cells, very few immune cells. There's a whole bunch of cancer cells that you see at 2 days only in mitosis. So the cell killing dynamics are slower in this immunogenic cell death, mitosis. And then we can also sort of just using existing scores, we can see there's a T cell cluster, there's a macrophage cluster, though there are some cells that seem to have signatures for both, and we'll get to that later, as well as NK cells and some endothelial cells. Okay, and broadly speaking, if we look at this response, we can see that there's a stronger T cell response in mitosis and a much stronger myeloid response in apoptosis. Okay. All right, so now we want to look at the T cell response. And if you have experience with this annotation of single cell RNA, you know this becomes complicated. So this is a phenograph clustering of the T cells from these different samples. They're all T cells, right? You can start looking at gene signatures, you can do marker gene analysis, you can look at differential analysis between clusters, and certainly some clusters are much more prevalent in the APAF1 knockout in mitosis, others are more prevalent in apoptosis. But is it, how to make sense of all of this? And just, again, not to give people nightmares if they have done this before, but there are a lot of markers you can look at, but this is a very time-consuming and uncertain way of labeling the clusters. So for example, you can see many clusters express PD-1, many clusters express granzyme B. Okay, so as an alternative, we turned in this project to topic modeling. And maybe for a computational audience, everyone's familiar with latent Dirichlet allocation and topic modeling in general. It comes from text, where we think of documents as having a distribution over topics, and the topics are shared across all the documents in the corpus. And each topic is a distribution over words. And we have priors that govern the peakiness of these distributions. So now we think of our documents are going to be cells, our words are genes, the topics are expression programs, coherent programs of genes that work together, and that will help us annotate. And we're not the first to use this, and it's not even our algorithm. We're using Kushal Dey's implementation, CountClust, that he developed for bulk RNA-seq, I think as a grad student, but works really nicely. Okay, so now we look at these T cells in immunogenic and non-immunogenic cell death, and we just do the topic models, and we find that there are 10 topics, okay? It's a Bayesian approach. You can sort of choose by Bayesian reasoning that there should be 10 topics. And the topics are actually very easy to annotate. So just as an example, topic 5 that we're calling a regulatory T cell program, if you look at the 30 top genes that are more discriminative of this, most discriminative of this topic versus all other topics, you can just see many genes you would recognize if you are familiar with regulatory T cells. Okay, so we found our immunology collaborators really loved this because it was just a quick way of understanding what the topics were. And on the right, what I'm showing is by cluster, and then just in apoptosis versus mitosis altogether, I'm showing the posterior probabilities of the topics. And on the over here, when you just look, you can see so more proliferation, more cell cycle, higher expression of the TCR components in mitosis, and sort of more of this OXPHOS program in apoptosis, less proliferation. Okay. You can actually do more, and I'll point out a few things. One is that you have doublets of myeloid and T cells that I think are coherent. I don't think they're artifacts. I think they're biologically meaningful doublets that are captured, okay? And in particular, you can see CD8 T cells with homeostatic macrophages or inflammatory macrophages, and you also see proliferating and non-proliferating CD8 T cells with mature dendritic cells, and those you see only in mitosis and immunogenic cell death. Okay, I also skipped the slide. This C8, which is this brown topic, is the effector cytolytic topic. You actually only see that in mitosis. Okay, so this slide is too much information. You know, as you know, I mentioned many more CD8 T cells in immunogenic cell death than non-immunogenic cell death, but we can say more. We can say what their states are. We can say that there's a broader range of activation states, including basically all the T cells with the cytolytic or effector program, mostly proliferating, not all. Also, the doublet, the dendritic cell CD8 doublets are all in the mitosis. Okay. In the apoptosis, it's more early and intermediate activation states and more non-proliferating. Okay. And you can also do the same thing for the myeloid component. I'm not going to present the details, and actually in the manuscript, we don't show all the topic analysis that informed these broad clusters. But what we can say is that there are more, you know, that the myeloid response is much stronger in apoptosis. So the myeloid cells are the sensors for apoptosis, but relatively speaking, there's an enrichment of these complement high monocytes and also CCR7 positive dendritic cells that home to the lymph node in immunogenic cell death. Okay, so last piece of this story. So I didn't show you a bunch of things that mitosis indeed generates a type 1 interferon response. The cancer cells secrete interferon beta, that in the knockout of cGAS, you reduce this kind of memory phenotype, don't eliminate it entirely, that it depends on CD4 and CD8 T cells. And the last thing is just, again, coming back to the motivation for cancer immunotherapy. The immune checkpoint blockade can synergize with immunogenic cell death. So what we're showing here is, again, the mouse experiments in the APAP1 sufficient and deficient melanoma cells in the mouse model. And this, these agents, one is a BCL2 inhibitor to inhibit an inhibitor of apoptosis, and the other agent enhances the first agent, the BCL2 inhibitor. And you can see that the drugs or anti-PD-1 alone have limited effect, but together they synergize and more of the mice live. Whereas in the APAP1 positive B cells, where you just get apoptosis, the mice all die. Okay. So with the time that I have left, I want to tell you quickly about two stories. I knew I have too much material. So just briefly, we have recently been pulled into these big patient studies where we profile the tumor immune microenvironment. So one collaborative study we did with Ari Hakimi and Tim Chan was an atlas of the tumor immune microenvironment in clear cell renal cell carcinoma, which is published last year. And you can see, you know, an atlas of 160,000 cells, and the lymphoid cells are here, the myeloid are here. You have some cancer cells and some stromal cells, and this nice annotation of interesting cell states. Okay, but to get there was a whole process of careful batch correction and annotation. And, you know, if you do this once for some set of patients, ideally you would want to learn a model and just annotate, automate the annotation, and deal with these batch effects through the model rather than by hand or by, you know, manual application of batch correction. Okay, so the idea is that we borrow a domain invariant variational autoencoder model that was first introduced for image analysis. And so the idea is that in autoencoders, you use so this is a deep learning method where you have an encoder and a decoder, and you're pushing the data through a bottleneck, a smaller latent space, and you're trying to reconstruct your original data. So you're forcing your bottleneck to learn important features of your input data. A variational autoencoder is a probabilistic version of this approach. It's a probabilistic latent model implemented through a deep learning framework. And in this DIVA approach, we want our latent spaces to disentangle the cell type information from the batch information. Okay, and we have, it's semi-supervised, so if it's your data set is partially labeled, so we have cell type labels, then we attach this classifier where we're trying to predict the known cell types. And we also encode the batches that we know in our data. Okay, and we have a batch classifier. Okay, so, and what we do, for example, training on our clear cell single cell atlas, we had, I believe, 29 samples from 6 patients, multi-regions of the tumor, adjacent normal, blood, lymph node, and we defined these 26 cell types, and we asked, can we annotate a completely held out patient? Can we annotate the single cells in these to these 26 immune cell types based on this DIVA approach? Okay, and just to give you an idea of why it should work at all, here we're looking at the cell type embedding at top and the batch embedding. So this is the bottleneck layer that's, you know, trying to disentangle cell type and batch. And you can see that the cell type embedding very nicely learns, you know, it has labels, it very nicely learns. Again, here the lymphoid, B cell, you know, a little to the side, and myeloid and cancer cells. And the cell types are well separated, but the batches are well mixed. Whereas in the batch embedding, the batches are well separated and the cell types are well mixed. Okay, and the accuracy seems competitive. So here I'm just showing on 2 data sets, our study and then a study from the next lab over at MSK, Donna Pear's group did this small cell lung cancer study, many patients, so more batches, but fewer immune cell types. And it seems to be competitive with SCNIM, which is the state-of-the-art deep learning approach for, and also semi-supervised for cell annotation. So we need to do a wider method comparison and a pan-cancer model, but this is where we're going with it. Okay, and if I have 2 more minutes, I want to show you one more thing, which is learning embeddings for single cell attack. Okay, so single cell attack, I think people are familiar, and it's been mentioned before in the meeting. It's really exciting, right? So you can resolve different cell populations and the differentiation states of cells in a heterogeneous sample. You can do single cell transcription factor analysis, for example, through ChromeVar, through sort of a motif accessibility score per cell. But most of the processing methods just borrow from single cell RNA. So instead of a cell by genes matrix, you start with a cell by peaks matrix, or maybe cells by genomic bins. It's very sparse, it's very high dimensional, and you have challenging batch effects. Okay, so for example, here I'm showing some data from Jason Buenrostro's paper. This is a bone marrow human hematopoietic data set. You can see how sparse it is. You can see that the dynamic range is 0, 1, and 2, okay, when you look at single cells. And if you do a standard kind of processing, a dimensionality reduction from Archer and nearest neighbor graph and UMAP, looks nice, looks like some structure, but you actually have some severe batch effects. So these hematopoietic stem cells that are being pulled into 2 regions, they're from different donors. Okay, so the idea of cell space, and I'm really going to finish soon, is that we use the sequence information by learning a common embedding of DNA k-mers and cells into the same latent space. Okay, and there's some important details of how we do this. We use the star space algorithm. We take peaks. We think of peaks as examples. We sample a sequence. We take the k-mers, and then we're embedding k-mers and cells, and we sample from the negative examples, lots of negative examples. We're embedding k-mers. It induces an embedding of this sampled peak sequence, and we're pushing it towards the cells in which it's open and away from the cells in which it's closed. And at the end of the day, we get an embedding into the same latent space of k-mers as well as cells. And we can also embed motifs after the fact. We don't have to start with a motif library, but after the fact, we can embed motifs into the same space and see the relationship between transcription factors and cells. Okay, so just a picture. We take that same data set, we use cell space, and here I'm showing the embedding of cells as well as the embedding of motifs that are relevant in hematopoiesis. And you can see that that bad batch effect is resolved, that all the hematopoietic stem cells from both donors are together, and that this hierarchy you can actually see in the embedding, and that if you start here and use Palantir, you can find all the most differentiated cell types represented. And one more data slide. You can overcome very challenging batch effects this way. So this is a bigger hematopoietic data set from Satpathy et al. They did a sort of bespoke batch correction, dimensionality reduction, found more variable peaks, did the dimensionality reduction again, and they got this structure. Okay, but without doing any of this work, just from the original data using a k-mer-based embedding, we can see a similar structure, okay, and separating the early hematopoietic cells from the more mature cells. So this is bone marrow and peripheral blood. If you want more information on these lymphocytes, you can separate them out and do a second cell space embedding just for them, and you can see that there's structure. Okay, so that's all I wanted to say. I want to thank everyone who did the work. Viraj did the cell death as well as the DIVA-based annotation. Zakia developed star space with help from Ali, and Ali Reza also contributed to the DIVA-based annotation project. And thanks Emily Cheng, Ming Li, and Sasha Rudensky for the first slide was a collaboration with Sasha. Okay, I'm gonna stop sharing if I can. Yes, and answer any questions if there's any time.
Yes, thank you very much, Christina. I think it was really great. I think we can do one or two questions if there are questions from the audience. If not, I can start with one of my many questions. I'm very curious about the topic modeling. So it seems like you have a combination of kind of genomic gene programs and also just cell types or cell states. Is it how it should be? And do you expect like for the cell type ones, for example, for regulatory T cells, they pretty much will not be kind of spread among many cells, but will be only for the regulatory T cells, and these cells also will not get any other topics?
So I won't.
I guess, of course.
Yeah, so I won't, go back to the slide because you need to move on. But you'll see, that regulatory T cell topic was only in one cluster, right? But then there's also the TCR components. There's that cluster has multiple topics, but in that case, some topics are highly associated with single cell types, and other topics are more generic, as you would expect, right? So the TCR components, it's across all the clusters to some extent because they're all T cells, right? So I think it is how it should be, right? That there's we want to, just like people regress that cell cycle, it's instead of regressing it out, model all the expression programs and see what happens.
Yeah. A question from the audience?
Yeah. Hello, Christina. Thanks a lot for the fascinating talk. I'm very interested in your DIVA model. And I mean, it's kind of like a chicken egg problem that anytime you have these semi-supervised models, is there any way that your model can deal with out of distribution cell types, like cell types that would only emerge in a novel donor? From what I could grasp from the slide, it will probably mix this. But is there a way from looking at the loss or any other that you could detect it possibly and correct it or annotate it afterward?
Yeah, I mean, I think that you have your classifier that's making assigning a confidence or probability of the label, and right now we're just assigning to the most probable label. But I think that one could do, you could threshold and say, I don't know, right? So I think that's an engineering detail that we need to do. We're trying to do a pan-cancer model where we cover all the bases and most things will be seen, but there can always in immunology be something that you should say, I don't know, or I don't recognize that cell type. So I agree, it's a practical issue that has to be addressed, and we haven't done that part, but we should. Yes, are you still there? Okay, so
could you hear the question?
No.
I'm sorry. The question is actually in the Q&A online.
Okay.
Have you applied topic models to single cell ataxic data?
The quick answer is no.
Okay.
Yeah.
That's good.
There is a cis topic, right? But the topics are just the peaks, right? And I don't think that's. I think you want the topics to be programs linked to transcription factor binding signals, right? It's not just that you want to represent cells by peaks, do topic modeling. I don't, so yeah. If that makes sense.
Well, thank you very much.